In this module, we will see how fatty acid enter into mitochondria via carnitine transporter. Fatty acyl-CoA ester formed. Fatty acyl-CoA ester is formed at the cytosolic membrane of the outer mitochondrial membrane. So this is cytosol and this part is matrix. This is inner mitochondrial membrane and this is outer mitochondrial membrane. And this space between outer mitochondrial membrane and inner mitochondrial membrane is known as intermembrane space. So first of all, fatty acyl CoA ester is formed at the cytosolic form of the outer membrane of mitochondria, and it can be transported into the mitochondria and oxidized to produce ATP, or they can be used in the cytosol to synthesize membrane lipid. Fatty acids destined for mitochondrial oxidation are transiently attached to the hydroxyl group of carnitine. So it is attached to the hydroxyl group of carnitine and it forms fatty acyl carnitine. It forms fatty acyl carnitine. This trans esterification is catalyzed by an enzyme which is known as carnitine acyltransferase 1. So this is carnitine acyltransferase 1 in the outer membrane. In the outer membrane. Now, the acyl-CoA passes through the outer membrane and it goes from intermembrane space to the inner mitochondrial membrane. Now, the acyl-CoA passes through the outer membrane and it is converted to the carnitine ester in the intermembrane space, or the carnitine ester is formed on the cytosolic face of the outer membrane, cytosolic face of the outer membrane. Then moved across the outer membrane to the intermembrane space. The current evidence does not reveal which. Passage into the intermembrane space occurs through large pores. It is the pore is made by the protein called porin in the outer membrane. The fatty acyl carnitine ester then enters the matrix by facilitated diffusion through the carnitine transporter of the inner mitochondrial membrane. In the third and final step of the carnitine shuttle, the fatty acyl group is enzymatically transferred from carnitine to intramitochondrial coenzyme by carnitine acyltransferase 2. So this is carnitine acyltransferase 2. This isozyme is located on the inner face of the inner mitochondrial membrane. It regenerates fatty acyl CoA and releases it along with free carnitine. So it releases a free carnitine. That carnitine into the matrix, carnitine re-enters the intermembrane space. So this free carnitine re-enters the intermembrane space via the acyl carnitine or carnitine transporter. So this is carnitine transporter. This three-step process for transferring fatty acid into the mitochondria, esterification to CoA, transesterification to carnitine, followed by transport and transesterification back to CoA. Links to separate pools of coenzyme A and of fatty acyl CoA, one in the cytosol, other in mitochondria. This pool have different function. CoA in the mitochondrial matrix is largely used in oxidative degradation of pyruvate, fatty acid, and some amino acid. But cytosolic CoA is used in the biosynthesis of fatty acids. So, fatty acyl CoA in the cytosolic pool, fatty acyl CoA in the cytosolic pool, it can be used for membrane lipid synthesis, or it can be moved into the mitochondrial matrix for oxidation. Mitochondrial matrix for oxidation and ATP production. Conversion to the carnitine ester commits the fatty acyl moiety to the oxidative fate. The carnitine mediated entry process is the rate limiting step for oxidation of fatty acid in mitochondria. So this is basically the way how fatty acid CoA enters into the matrix. Let's recall the main things again. So this part is cytosol, this is intermembrane space, and this part is matrix. And this membrane is outer mitochondrial membrane. This membrane is inner mitochondrial membrane. Here is fatty acid CoA. It attaches with CoA SH by the help of carnitine acyltransferase 1. Then it goes in the intermembrane space and it goes to the matrix through a transporter, and then carnitine separates carnitine free carnitine, then it reaction is done by carnitine acyltransferase 2. Now the free carnitine moves in the intermembrane space with the help of transporter. Then it again comes to the cytosol. Then this process continues. So this is the way how fatty acid, CoA or fatty acid enters into mitochondria by carnitine transporter.